So viruses. So viruses, we do not consider them as living organisms. So because they do not fulfill some of the basic parameter or basic characteristics which a living organism should possess. That means if you want to consider anything as a living organism, it should go, first thing is it should show a growth. So the growth which should be increasing, the increase in the size, cell size or organism size from within, not from outside. So even the inert material like the wooden sticks or the rocks, they show increase in size by deposition of the dirt outside. So outside growth, we do not call it as a growth. It should grow from inside. From inside, it should multiply or become bigger. Second one is it should show metabolism. It should have a metabolism. That means it should take up the food inside, it should metabolize and it should excrete. Okay. And third one is multiplication. It should reproduce or it should increase in size, or all the organisms, they should reproduce, they should multiply. So out of these three characteristics, so viruses shows only one character. It doesn't have a cell. It doesn't contain a cell itself. They do not have a cell wall, cell membrane, cytoplasm, cell organelle, nothing. They do not possess a cell itself. Second one is they do not show any metabolism. They do not take the nutrient inside. So they do not have a metabolism. They do not excrete. Third one is outside the outside the host is when they are present freely in the environment. Viruses do not show any growth, they do not show any movement, nothing. So they are like any other inert materials like rock, dust, and other things. They don't show any form of growth. Only when they are present inside a host, they show only one parameter of growth, that is multiplication. They multiply only when they are present inside the host. So they do not show any type of growth or metabolism. So when they are present outside the host. Okay. So now we can call viruses as acellular obligate intracellular parasite consisting of a genetic material surrounded by a protein coat. So all the viruses, they have a two main component. One is a genetic material, either made up of DNA or RNA, either single stranded DNA or double stranded DNA. And they have a covering which is covered by a protein coat. And they are obligate means the requirement of a host is obligate necessary for their multiplication. So they are obligate intracellular parasites. So therefore they define viruses as acellular obligate intracellular parasite consisting of genetic material surrounded by a protein coat. Okay. And some of the characteristics of viruses are they are acellular, living particle. They do not have any cell system. No cell membrane, no cell wall, no cytoplasm, no cell organelles. So they have a genetic material which is made up of either DNA or RNA. They are obligate intracellular parasite. That means to say that they show growth or multiplication. They do not show growth, they show multiplication only when they are present inside a host. It is incapable of independent growth in an artificial media. It cannot be grown in an artificial media in laboratory. And coming to the host, which are all the host or which are all the other living organism, it will infect and cause diseases. So please remember, so virus infects all other organisms present on the earth. It infects all the living organisms that are present on the plants. It infects viruses, it infects fungi, it infects algae, it infects plants, it infects animals, it infects human beings. So it causes a diseases on all the living organisms that are present in the earth, are present on the earth. Reproduction, coming to the reproduction, viruses do not show growth and production when they do not show growth or they do not produce spores, anything when they are present outside a host. Only when they are inside a host, So they do a reproduction. That too, there is multiplication. So multiplication is nothing but just they replicate their genetic material. So they produce protein coat and they pack the genetic material in protein coat and come out of the host cell by breaking the host cell. That's all. They multiply only when they are present inside the host. And they do not carry out any metabolism. Size. Coming to the size of the viruses, they are very, very small. These are the viruses are the smallest, to say they are not living organisms. To some extent, since they multiply inside the host, they are the smaller, they are very smallest compared to all other organisms. So their size vary from 20 to 30 nanometer, not micrometer, 20 to 30, I'm sorry, 20 to 300 nanometer in size. The largest virus is 300 nanometer, so which is equivalent to the smallest bacteria. So, and their genetic material vary from 2 kilobytes to 4 megabytes, 2 kb to 4 MB. And they can pass, since they are very small, they can pass through the pores or the filters, so which are used to filter bacteria. The bacterial filter cannot hold the viruses since they are very small. And they are not visible to naked eye. They cannot be seen through naked eye like other microorganisms, nor, and even they cannot be seen through the compound microscope, so which can give a magnification of 1000 to 1500 times. So with the compound microscope, we cannot detect the viruses because they are too small. And they are visible only under electronic microscope. So they are visible only under electron microscope. Both scanning electron as well as transmission electron microscope can able to see the viruses, where you will get a magnification of at least minimum of 1 lakh time. Coming to the shapes of the viruses. So these are the some of the shapes of general viruses that are present. This is called as an icosahedral. Icosahedral means it is as a many faces. Or we can say it is a polyhedral. In polyhedral, icosahedral means it is a polyhedral with 20 faces. Polyhedral with 20 faces is called as a icosahedral. So this is the below one, this is called as a helical. You might have heard about this. So tobacco mosaic virus, you might have studied about that. That is a helical initially. And these, there is another viruses which are called as envelope. Along with the genetic material and protein coat, this genetic material and protein coat is covered by another layer of a coat, which is called as a envelope. So icosahedral envelope, here helical envelope. And there is other viruses, so which are called as complex. They do not have a proper shape. They have varied shapes. It is called as a complex. So icosahedral, helical, envelope, and complex viruses. So these are the major types. Here we can see this is an helical, this is polyhedral, which I call as a icosahedral, 20 faces. This is a spherical. So the spherical was not mentioned here. So spherical, enveloped, spherical. I'm sorry, this is spherical because there is an envelope. Helical viruses is enveloped by another layer because of which it is becoming spherical, like HIV virus or coronavirus. And this is a complex virus. Example is bacteriophages. Here they have given an example. Helical, tobacco mosaic virus, polyhedral adenovirus, spherical or enveloped virus, influenza virus, complex virus, bacteriophage. Same thing here, viral shapes. Another slide. Polyhedral, spherical. Spherical, this is because it is an envelope. Complex and helical. What are the characteristics of viruses? This you may remember. What are the characteristics of viruses that differentiate them from microbes or other living organisms? So this I have already told you their character. If you remember their characteristics, you will come to know. So they lack fundamental structure of a living organism, that is cell. They do not have cell and they do not have cytoplasm and they have no independent life support system. They cannot show growth independently. They have no means of independent life support activity. They do not have enzyme. They do not carry out metabolism like that. So they lack basic functions of life, like they lack basic functions of life, like metabolism. They lack metabolism, they lack reproduction, and they lack adaptation. And as I said, the viruses do not carry out metabolism. They cannot metabolize nutrients, and they cannot take up the nutrient. They cannot metabolize and excrete the waste. So, and they cannot move their own, and they cannot reproduce their own, except when they are inside the host cell. So they rely entirely on the metabolic activity of the host cell. Even the enzymes required for its multiplication, and even the enzymes required for its replication of its genetic material is taken or utilized by the host cell. Okay. Next year, you should remember some of the terminologies. They may ask it for one mark questions, so which you need to remember. First one is virion. So virion is nothing but a virus particle. It is a mature virus particle is consisting of a genetic material surrounded by a protein coat or capsid is called as a virion. Mature viral particle which is ready for infection, which is containing a genetic material covered by a protein coat is called as a virion. There is another one which is called as a viroid. Virion and viroid. Viroid is nothing but it is just a genetic material which is infective. A naked infective RNA or small circular RNA molecules which possess no protein coat. So protein coat is also called as a capsid. So a only genetic material or a RNA particle which is capable of causing disease in plants is called as a viroid. Okay. And capsid, the protein coat or the shell that surrounds the genetic material of a virus is called as a capsid. And capsomere, the individual protein units which make up the protein coat or a capsid is called as a capsomeres. Example, I'll show some photo. Here we can see this whole thing is called as a capsid, the protein coat. This individual protein unit which make up the capsid, they are called as a capsomeres. Okay. Next, coming to the discovery of viruses. So actually, first bacteriophages were discovered by a scientist called Frederick Todd in 1955 and Felix D. Erel. Felix D. Erel and Frederick Todd discovered independently the bacteriophages, so which are viruses in 1915 and 1917 respectively. And what are bacteriophages? So I said that viruses infect all other organisms. The viruses which will infect bacteria are known as bacteriophages. Bacteriophages are also known as phages. These are the viruses that infect bacteria. Okay. So here you need to study about the bacteriophages and their life cycle as per your syllabus. So now we will discuss on the structure of bacteriophages, characteristics of bacteriophages, structure of bacteriophages, and their life cycle. These are the characteristics of bacteriophages in general. So the bacterial viruses are widely distributed in nature. They are present everywhere. All the environment like soil, water, air, oceans, everywhere. And the phages are specific to particular bacteria, and bacteriophages are present for all the types of bacteria. That means to say that there are different types of viruses which will infect different types of bacterial cells. So with proper techniques, these bacteriophages can be isolated in laboratory. I said that the viruses cannot be cultured in laboratory, but with some techniques, we can isolate, we cannot grow. Even we can grow with some technique in the laboratory that I will, whenever there is any free time, we will discuss it. Like other viruses, so bacteriophages are also composed of nucleic acid surrounded by a protein coat. And bacterial viruses bacteriophages also comes in different shapes. And many phages have a tail through which they inoculate the genome host. So although there is a particular shape for a bacteriophage, there are also some variation in the bacteriophages. Some have a tails, some do not have a tails. Like other viruses, bacteriophages have either single-stranded DNA or double-stranded DNA or RNA as a genetic material. Bacteriophages may have single-stranded or double-stranded DNA or RNA as a genetic material. And most of the bacteriophages, they have a double-stranded DNA. Most of the bacteriophages, they have a double-stranded DNA as their genome. And few bacterial viruses, apart from protein, they may also have a lipid envelope, like capsid, which is made up of a lipid. However, many phages are structurally complex. Structurally, compared to other viruses, they are a little bit complex in nature. They do not have a proper shape, proper structure. So here you can see the different shapes or structures of a bacteriophage, which will infect different types of organs. Mycoviridae, which will infect, I think, fungi. This is a bacteria infecting viruses, all others. So you can see the different shapes here. So you need not have to worry about this. Coming to the structure of a bacteriophage. So all the bacteriophage, as I said, they have a nucleic acid. They have a nucleic acid surrounded by a protein coat. All the bacteriophages. But other structure may be present in, as I say, I am showing here. So other structure may be present or may not be present. But this structure, basic unit, genetic material and protein coat will be present in all the bacteriophages. This is a T2 bacteriophage. This is a one bacteriophage which is called as a T2 bacteriophage. This is the structure of this T2 bacteriophage. So in general, if they ask you to write a structure of a bacterial phage or a T2 bacterial phage or a T bacterial phage, you can write this structure. So this is a genetic material which is a double-stranded DNA. This is a protein coat which is icosahedral. And this is the collar. This is the collar. This is called the sheath. This is the sheath. So this is the base plate. This first plate is called as a collar and second one is a base plate. So base plate contains hook-like material. These are called as hooks or spikes. And this base plate is also fitted with a tile fiber. Tile fiber. Okay. Genetic material, capsid head, collar, base plate. This is sheath. These are hooks or spikes and these are the tile fibers. And coming to the life cycle of bacteriophages shows two types of infection in bacteria. Bacteriophages show two types of infection or a life cycle in bacteria. One is called as a lytic or virulent phase. It is called as so it shows a lytic cycle. Another one is called as a temperate or avirulent phase. It shows a lysogenic cycle. There are two life cycles of a bacteriophage in bacteria. One is called as a lytic phase or lytic cycle. Second one is avirulent or temperate phase, temperate cycle or a lysogenic cycle. Lytic and lysogenic. I will explain you what will happen in lytic cycle and what will happen in lysogenic cycle. In general, majority of the bacteria shows lytic cycle where they are highly virulent. They will kill the bacterial cell. So this is the general cycle of a bacteriophage. So general cycle of a bacteriophage, which is for the majority of the bacteriophages, which shows the lytic phase. This is for a lytic phase because majority of the bacteria shows virulent or a lytic phase. So life cycle of a bacteriophage consists of 5 stages or 5 steps. Okay, one is attachment or adsorption. So the bacteriophage goes and attaches onto the bacteria. Second one is, so it makes a penetration and injection. It will go and sit on the bacterial cell wall. It produces the lysozyme enzyme. It dissolves the bacterial cell wall. It makes a hole by punching through hooks or the sheath that is present on the base plate. So then by pressing on the sheath, the sheath is like a spring. So by pressing on this, with the help of this holding, with the help of tail fibers, it will inject the genetic material inside the bacteria. First one is attachment and adsorption. Second one is penetration or injection of the genetic material onto the bacterial cell. Third one is synthesis of nucleic acid and protein code. So it will synthesize its protein, nucleic acid and protein code. Next, it will assemble the protein nucleic acid in its newly formed protein codes. Then it will break the cell wall of the host cell and it will come out. So attachment or adsorption, penetration or injection, synthesis of its nucleic acid and protein coat, assembly of nucleic acid in its protein coat and packing, and release of the mature virions from the cell. These are the 5 stages. Same thing here. Same thing, life cycle and lytic phase. In lytic phase, what will happen is, so the bacteria will kill the bacteria and comes out. It will kill the bacteria, it comes out. What will happen is, the phage attaches by its tail fiber to the host cell. It will penetrate, it will dissolve the cell wall and it will penetrate its genetic material into the bacterial cell. The next one is biosynthesis. These are the steps in lytic life cycle of a bacteriophage. Attachment, penetration, biosynthesis. It produces its genetic material and proteins. Maturation, it assembles the bacterial DNA in its protein code and release. It lies the cell wall, break down the cell wall, and the active virions or viruses will come out. This is the life cycle of a lytic phase. Here again, once again, I am explaining. So initially, adsorption and attachment, the bacteriophages will come and sit on the cell wall of a bacteria. So in the second one, injection of nucleic acid, it dissolves the cell wall by producing lysogenic enzymes. With the sheath or hook, it will make a puncture. So then by pressing the sheath, it will release genetic material into the host genetic material. So next one is biosynthesis. So once the genetic material enters inside the bacteria, it takes over the control machinery of the bacterial genetic material. So it uses bacterial genetic material to synthesize its own genetic material and protein code. So then assembly, the production of it produces its own genetic material and protein code, and it assembles its genetic material in its protein code. It breaks down the cell wall and comes out. So in lytic phase, the bacteria enters, synthesizes its material, packs, kills the bacteria, and comes out. This is a simple life cycle of a lytic or a virulent bacteriophage. And another one is lysogenic. There is one more phase, temperate cycle or a lysogenic life cycle. So here what will happen is, so once the genetic material is pumped into the host cell, this genetic material of RNA, it goes and binds to it joins the host genetic material. And it will not do anything to the host. It will not harm. And it will keep on replicating. And each binary fission, whenever bacteria replicates, so this virus also replicates and moves to daughter cells. That's all. So this phase is called as a lysogenic phase or temperate phase. So where the genetic material of virus will be integrated into the host genetic material. When genetic material of the virus is integrated into the host genetic material, this condition is called as a prophase. So prophase, and bacteria is inactive. It is not virulent. It is inactive. So in the host, and it just keep on multiplying along with the bacterial genetic material, and it can goes on with the new daughter cells. It will be multiplying and it will be passing on to new cells. That's all. It will not harm the bacterial cell. It will not kill the bacterial cells in lysogenic or a temperate phase. But over the time, many cells contain a bacteria. Okay. But with the time, and this is not a continuous, this is not, and this temperate phase will not be permanent. So with some time, so what will happen? This genetic material which is integrated in the host cell may comes out. It may comes out of the integration. From prophage, it may come out outside the genetic material and it may convert it into lytic phase. It may this virus may convert into lytic phase where it will take the machinery of the host genetic material, it multiplies its own protein coated genetic material, packs, kills the cell and comes out. Huh? Okay, this is about lytic and lysogenic phase of the viruses, and this is about viruses. So this is what I wanted to cover in this class. If anybody has any doubts, you are free to ask.